In this video, we'll talk about the phases of T-cell mediated immune response. If we plot the immune response in y-axis and time in x-axis, then we can visualize the events of T-cell responses in real time. So the first phase is activation phase, where the T-cells interact with antigen presenting cells and get activated. The second phase is clonal expansion, the third phase is effector response phase, and then eventually the number of T-cell would decline. So there is a decline phase, and lastly memory T cells might be generated. So it's like a memory phase. So all these 5 phases would be discussed in this video step by step. So stay tuned till the end. So the first step is activation phase. Dendritic cell or any other antigen presenting cell would recognize a pathogen associated molecular pattern or PAMP using the toll-like receptor present on their surface. After recognizing this, they would display this antigen on class 2 MHC molecule and migrate towards the lymph node. And now the dendritic cell is in the lymph node. In the lymph node, it would interact with naive T cells with the help of TCR and CD4 co-receptor. This interaction gives rise to the signal 1 for T cell activation. B cell expresses a molecule known as CD80 and CD86, and T cell expresses CD28. This specific interaction gives rise to the signal 2 for activation. And the third signal for activation comes from the cytokines. In context of T helper cells, IL-2 is the key cytokine that gives rise to the signal 3. All these 3 signals lead to the activation of T cell. What happens when the T cell is activated? Generally, the naive T cells are resting in the G0 state. They are quiescent. After activation, they would re-enter the cell cycle and proliferate rapidly. This is known as clonal expansion. It is important to note that out of many different types of T helper cell or CD4 positive T helper cells, only the cell which is specific for this antigen would clonally expand, not all of them. Only a selective population would get expanded. This is known as clonal expansion. And clonal expansion of lymphocyte is a hallmark for vertebrate adaptive immune responses. Now there would be an effector phase where the T cells would interact with B cells. So this is the CD4 T helper cells. Eventually they would activate B cell into plasma cell, and plasma cell would secrete antibody to show the responses. So they coordinate with B cells. Also they can secrete cytokines which can attract many other cells including APCs, neutrophils and many immune cells. On the other spectrum of the effective response, there is CD8 positive T cells, which are cytotoxic T cells, which secrete cytotoxic granule and kill the cells. In the second half, we would talk about the cytotoxic granule and the effector response for CD8 positive cells. Now let's get back to the CD4 positive cells. CD4 positive T helper cells are generally helper cells. So they secrete messengers that can trigger other cells, and they can do many jobs indirectly. So there are many subtypes of these T helper cells, and each of these subtypes secretes different type of cytokines, such as Th1 positive subtype secretes interferon gamma and TNF. Th2 secretes interleukin 4, 5 and interleukin 13, which are important for dedicated functions. T regulatory cell secretes anti-inflammatory cytokine like interleukin 10 and TGF beta. Th17 secretes pro-inflammatory cytokines such as IL-17A, 17F and IL-22. So all these cytokines has their own immune modulatory effects. Now another important job of T helper cell is to activate the B cell. T cell interacts with the B cell with specific T cell receptors, and there is a interaction of CD40 and CD40 ligand which leads to the B cell activation. Activated B cell would proliferate rapidly and expand clonally. Activated B cell in the lymph node would undergo processes like affinity maturation, which selects the high affinity antibodies on the B cell surface. Also, they would undergo a process known as class switching or isotype switching, which would produce a specific type of antibody and from the plasma cells. So eventually the activated B cell would differentiate into the antibody secreting plasma cells. Now we are coming back to the cytotoxic CD8 positive T cell responses. Imagine this yellow cell is now affected by the virus. So once the virus has entered the cell, some amount of the viral peptide would be also displayed on the class 1 MHC molecules, And this class 1 MHC molecule bound peptides can be recognized by the CD8 positive cytotoxic T cells. Immediately, these cells know there is something wrong, so they have to act promptly. They secrete several cytotoxic granules which has perforin and granzymes. These particular cytotoxic granules get into the cells, so perforin and granzyme lead to activation of apoptotic pathway. Granzyme activates bead on the mitochondrial membrane, which allow the leakage of cytochrome C. Once cytochrome C is leaked in the cytoplasm, it can interact with APAF1, eventually leading to the activation or cleavage of inactive caspase 3. Cleaved caspase 3 is active and can execute the process of apoptosis. There could be also FAS-L and FAS mediated apoptotic pathway, which lead to the activation of caspase 8, and eventually the executor caspase 3 is activated. As a result of these caspase activation, death of the virus infected cell is caused, and the nearby cells are saved from the viral infection. This is how a CD8 positive T cell work, and this is the effector mechanism. Be it CD4 positive or CD8 positive T cell, after the job is done of pathogen, so when the pathogen elimination job is done, so many cells doesn't need to be there. So they have to be eliminated, and immune system selectively eliminate them via apoptosis. So there are few cells which are remaining. These cells would eventually become the memory T cells. When there is a second time of infection, these cells would promptly act in the second round of infection. Now it's also important to understand the antigen exposure kinetics determine the response kinetics. So here the T cell response kinetics is in red, and blue denotes the antigen exposure kinetics. So here is the T cell response kinetics, where you have a naive T cell which would be activated and it would ultimately become T effector cell, and eventually when the pathogen is eliminated, antigen level is down, then it would become a memory T cell. This is the normal scenario, but in many cases T cells get exhausted when they encounter pathogen for a very long time or antigen for a very long time. They get exhausted, they become unresponsive towards stimulation, and this is known as a state which is characterized by specific molecular markers. This is very important in context of cancer. CD8 positive T cells fight the tumor cells and they eventually kill the tumor cells. But once they are exposed to tumor antigens for a very long time, because tumor cells are dividing at a very rapid pace, then the CD8 positive T cells cannot cope up. They eventually becomes very exhausted and go to a state of unresponsiveness. They are characterized by expression of several molecules. It's a hypofunctional state, and the efficiency of these cells are dramatically compromised. At this kind of scenario, there are two options: either fight till the last breath and die, or the second option is fight till the end but now become unresponsive. So basically option B is selected by many cells because they get tired, they get unresponsive, but it's a reversible state. Eventually, with proper time and proper stimulation, it can be reversed. But these are the new findings these days in T cell research. I hope this video was useful and we can appreciate the T cell response kinetics. If you need more notes and flashcards, quickly visit our Facebook page and Instagram. You can support us using Super Thanks. You can pay via PayPal, Paytm and UPI. See you in next video.